Welcome to Unit 3. For this unit, we will focus on the exciting field of genomics. This is a very exciting time for genes and genomics as we have advanced or harnessed new technologies that allow us to take a look at the genomes of more organisms quicker, cheaper than ever before. And the amount of new data that is coming out of the field of genomics is absolutely extraordinary. You are probably already familiar with these folks here. This is Watson and Crick. These guys won the Nobel Prize for determining the structure of DNA, although they are often also credited falsely with discovering DNA. And so I thought it important for us to take a quick walk through a little bit of history of the genomic sciences so that we can learn a few of the players that were involved, the key figures involved in making incremental discoveries that ultimately led to the discovery of the structure of DNA. Many of you probably heard or learned about Gregor Mendel in your high school biology courses. Gregor Mendel, of course, was the scientist back in the 1820s. Actually, he was born in 1822, but did his work in his 30s, so more around 1850, where he did the breeding experiments with pea plants. And this was sort of the first historical experiments that were done to try to figure out how an individual organism, in this case plants, were obtaining their physical traits. And so Gregor Mendel had several different varieties of pea plants that he crossed over several different generations and observed that these traits were being passed on from generation to generation and that some traits were passed on more often than others. Moving ahead, though, to 1860, this gentleman here, Friedrich Miescher, is really credited with being the individual who discovers DNA. And by isolating a material that he named nuclein from inside the nucleus of white blood cells, he actually found DNA. Now, Friedrich Miescher is often not credited with that discovery and is left out of the historical records, but deserves that recognition nonetheless. The next major player in the history of genomic science is Phoebus Levine, and Levine was a Russian-American scientist. He studied the nucleotides, or the individual components that make up DNA and RNA, extensively. And Levine set forth what he called a tetranucleotide hypothesis. He was an arduous scientist. He worked diligently to try to better understand the composition and the makeup of DNA. He also studied or tried to study extensively and put forth different theories on the structure of DNA. He was an extremely well-published scientist, over 700 articles credited to him, but he never did get the structure quite right. And so his tetranucleotide hypothesis, and you see it here as the image on the right, is indeed incorrect, as we now know, but it did sort of lend to his knowledge on the nucleotides, enabled Watson and Crick to develop some of the background and ideas that they needed in order to come up with the structure of DNA. Moving along, Erwin Chargaff in 1950 also studied extensively the genomes and DNA of several different organisms. And what he noticed is that there was a mathematical formula that could be derived by looking at the base pairing or the numbers of different purines and pyrimidines found in the DNA of the different organisms. And so he found if you added up all of the purines, those being the A's, the adenines, and the guanine nucleotides, that always equaled the amount of pyrimidines found in the organism. So A's plus G's equaled T's plus C's. And this is known as Chargaff's rule. And this, again, was another piece of evidence that Watson and Crick relied heavily upon when trying to decipher the structure of DNA. The next sort of major player in history is Rosalind Franklin. Now, at around the same time that Watson and Crick were trying to determine the structure of DNA, Rosalind Franklin was also working quite diligently on this effort. And so she had studied through her training, extensively worked with X-ray diffraction or X-ray crystallography. And it is actually this image on the right. This was part of her lab work. This was an X-ray diffractory image of DNA that Crick and Watson used to come up with their theory as to the structure of DNA. Now, there's a little bit of a scientific scandal here because Rosalind Franklin was not awarded a Nobel Prize along with Watson and Crick and Wilkins. She was left out of that recognition. And her data here, Crick later admitted, was sort of the key piece of evidence that they ultimately needed and used to come up with their structure. Dr. Franklin here, or Miss Franklin, I should say, died very young at the age of 38 of ovarian cancer. And there's, of course, a lot of scandal and sort of insinuation that perhaps this was sexism. Being a woman at the time, particularly a woman scientist, she didn't deserve gain the recognition that she deserved. So, scandal aside, we can all agree, though, that she made a major contribution to the field of science. Her work here was certainly instrumental in determining the structure of DNA, and she certainly should receive a place in history for her efforts. And lastly, we come to Watson and Crick, and there in 1953, they put forth and published this proposed structure. Here you see this double helix with the turn in it. This, in essence, allowed them to incorporate all of the pieces of data that had come before them. Franklin's data on the X-ray diffractory, Chargaff's rule showing that you needed to have the particular base pair matches. And so they were ultimately awarded the Nobel Prize for their discovery of this particular structure. If we continue to move forward then, and fast forward now to about the late 1970s, the scientists became very interested in determining or being able to fully sequence then or determine the individual nucleotide components of different pieces of DNA. And so Fred Sanger put forth this dideoxynucleotide chain termination sequencing methodology in the 1970s. This allowed for scientists to determine the individual nucleotides in order for a particular piece of DNA and became sort of the sequencing method of choice for many, many years. And Sanger sequencing is actually still used extensively today. But if we fast forward to modern times, and in the past 15 years or so, there's been an explosion of new sequencing technologies that allow us to determine the nucleotide sequence of a particular genome or piece of DNA much quicker and much cheaper than ever before. And two examples listed here are 454 pyro sequencing and an Illumina technology. Both have the same outcome in essence, but use slightly different technologies. There are advantages and disadvantages for each. And these are just two of several that are now available commercially on the market for scientists to use in determining a DNA sequence. So with the explosion of new data that we have, many different disciplines have been affected. And you're probably already aware of the changes that have come in the criminal justice system as a result of DNA sequencing. It plays a major role now in many different cases. But other areas, particularly in the sciences, have also been dramatically affected as well. And this new discipline developed as a result of all of the bioinformatics information, and this is a pharmacogenomics. And this is the idea that you can take an individual sequence, or right now the field is really at a population-based sequencing level, and design drugs specifically for different diseases within different populations. And the goal is really to get to the point where we could take genetic information from a particular individual and have some sort of sense of how this drug will work in that individual before even administering it to that person. Now, you can begin to imagine some of the difficulties and ethical issues that arise with this type of science, but it is something that may be possible in the future and is already being studied extensively today. In addition, the great amount of genetic information plays a role in treating and recognizing diseases very early. You have genetic screening that can occur. We've started to touch on some of this in our stem cell unit, I should say, and our newborn screening issues or unit. Furthermore, then the pharmaceutical industry, again, is using this new genetic information to try to design drugs on the front end that will be more effective, work faster, target specific regions, target specific genes, to be more effective for treating a variety of things, anywhere from targeting specific viral infections to better treatments for cancer, etc. And so this whole explosion of data has also led to a new field in bioinformatics, and this is an extremely quickly growing discipline in which computer technologies are extremely important for deciphering the sheer volume of data that is coming off of these sequencing machines. Now, this marks the end of our introduction and a little bit of the history of genomics. The next short video clip I included because it gave a really nice representation of how DNA is operating in the cells to make message RNA and then ultimately proteins for normal cellular functions. I liked the video because it gave you a sense, or it gives you an idea of how these things are working together in the cell in a slightly different dimension than just simply looking at basic cartoons from a textbook. And so do not be alarmed. You don't have to really understand everything that they're talking about right now. You can sort of sit back and enjoy the visual tour. We will cover all of these things in greater detail in the different lectures throughout the unit. And so I hope you enjoy the unit on genomics. I find this field particularly fascinating, and it really is an emerging field where information is changing every day.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.